disconnection?
What is the disconnection?
You are seeing the expression.
You are seeing the expression.
Whose expression you were
Whose
checking?
expression you were
Go
checking?
back to the previous
Go
slide.
back to the previous slide.
So here you are seeing over.
So here you are seeing over expression because your aim was to
overexpress
the gene. Now
in the next one, whose expression?
You are saying HL1.
Here,
Why reporter came?
You didn't explain where
You
the
didn't
reporter
explain
is.
where the reporter is.
Huh?
From where you brought this reporter in this story?
So actually, we can check this. We can check the guide RNA that we are targeting, which we are making for our promoter, whether it is working or not. We take the promoter of our gene along with the reporter, guide RNA.
Understood.
Yeah. That is the difference.
So disconnect. So when you are saying that we are going to do this work, we are going to overexpress, we'll check the expression. Fine. Because that is the target. Our aim. Here suddenly this is you have changed the gear. Suddenly you from first gear to go to third gear, right?
And we wonder why it is happening.
So this is the disconnect.
So this is
either
the
you have to show that you will see it as a model,
see it as a model.
In some plant system,
In some
you
plant
put
system, you put reporter
and you do that
overexpression thing
with
in a
reporter gene.
Check it whether it is
Check
working
it
or
whether it is
not,
working
and
or not, and then you move to
the main system.
AHL1.
Or
there is no need for this.
There is no need for this.
If you are seeing an
If you are seeing an
overexpression, yeah, but that you can do with HL1.
Yes.
So these are two different. So there is no
flow in your
methodology. So that you have to see. Suddenly you are doing something and you shift to
So
something
this
else.
can be a different
In fact,
experiment.
this can be the first experiment
To see a proof of concept
whether your
technology, what you are planning,
strategy is working,
and then you
are moving to
rise and
with the proper gene,
which is your target.
So there is a
disconnect here.
Okay, next one.
This is all fine. Good.
Thank you.
नहीं प्रेजेंटेशन
इनके पास यहाँ पे लगाना है क्या
a very warm good morning to everyone. Good morning to everyone. All the over here and my fellow training program mates. So as the topic you can see it is being flashed on the screen. It is genome editing assisted breeding for development of basmati rice varieties. So India is the biggest exporter of basmati rice across the globe. Around 95% of the rice is being consumed across by humans only. In that 95%, 75% is basmati rice. So that's why we literally focused on this particular trait of basmati rice. So as you can see, I'll not go into that what rice is and whatever. Our major aim is to prepare a particular basmati rice for dry DSR condition. What is DSR? DSR is your direct seeded rice where we are going to skip that particular part of puddle. As you can see that I have mentioned there are like a major constraint in rice production. And in that particular thing, what you're pricing is what you're pricing dry DSR. What is DSR? The consumption of water is human rice where we are going to skip that rice production is one. As you can see that I have mentioned there are major which we want to skip and format. In that what you're pricing is what you're pricing is dry DSR. What is DSR? The consumption of water is human rice where we are going to skip rice production is one. As you can see that I have mentioned our chances that we are going to lose that particular aromatic. So that aromatic content we are going to plant it across different locations. And if it's coming up to market buying basmati rice for activities like the minimal. So do you think that that's not going to combat that aromatic portion? We are actually resolve this particular problem by combining marker seeding along with the genome editing portion. So rice is because of a volatile compound which is 2-acetyl 1-pyrrolene which is 2-AP, and it is actually a key component, but it is having a very strong inhibitor which is bad H2. As the name suggests, it is doing some bad activity and it is curbing the presence of 2-AP which is giving the fragrance to rice. And if we are going to come up on that particular ground of dry DSR basmati variety, we can combat biotic factors as well as abiotic factor. But this particular factor which is aroma, we can't combat it by conventional molecular breeding. So I'm going to hand over podium to my fellow mate.
So we have selected this gene because it is a negative regulator. So we have selected BATH2 gene because it is a negative regulator for aromatic compound that is 2-acetyl 1-pyrrolene. So it is responsible for fragrance in rice. We have chosen this to knock out this gene. And BADH2 gene to breed for quality improved rice cultivators for direct seeded rice.
Let us know regarding the pathway that is responsible for 2-acetyl 1-pyrrolene compound. As Sakshi mentioned, during DSR condition, there will be a reduction in the fragrance content because of reduction in the formation of 2-acetyl 1-pyrrolene compound. From this pathway, you can see that the glucose compound is converted into methyl glycosol, which is converted into 2-acetyl 1-pyrrolene component. During stress condition, that is during direct seeded rice condition, the methyl glycosol content will be more. It means it should produce the 2-acetyl 1-pyrrolene content in enormous production. But because of overexpression of the functional BADH2 gene, there will be a reduction in the 2-acetyl-1-pyrroline component. As a result, there is no fragrance content or aroma in our basmati rice during direct seeded rice. So we thought of knocking out this functional BADH2 gene in order to get the over-acetyl 1-pyrroline component in direct seeded rice condition. As a result, three components interconnected to acetyl 1-pyrroline compound. One is glutamate converted into glutamate gamma semihaldehyde pyrroline 5-carboxylase which results in formation of pyrroline, which in turn gamma aminobutylaldehyde, and then gamma aminobutylaldehyde is converted into GABA because of functional BADH2 gene. We can see this. Here is the key important point where we need to knock out the BADH2 gene, which in turn results in the accumulation of pyrroline, which is a precursor for formation of 1-pyrroline compound. So we thought of taking or knocking this gene for our project proposal. I hand over this to Akshay.
So now we'll be talking about this OSBA, and when we look at the gene structure, we can see that there are 15 exons present in this. Out of these 15 genes, the basic strategy to make the knockout is to initial terminal, so that we can get mutation like that in the further genetic strand, in the DNA strand, where we can get the whole protein that is forming. That we can see that there are multiple binding sites and active sites are present in here. 503 translation residues, and the protein that is forming after this knockout in the initial form a non-functional one. Because of this, we can target this initial regions, and then later on I check for different guide RNAs in the initial exons. Then I found that for the third exon there is one guide which was showing the on score of 30%, and there was some of the off targeting score which was about 0% in the exonic region. So if this target intergenic region, then there won't be any issue for our gene editing. We were targeting only the exonic region. Another thing is like we also found another guide RNA in the fourth exon, where we see that there is around 40% of on score efficiency, and there is no exonic off targeting here. Then considering these two are the guide RNAs for our genome editing, we found that because of the very less knowledge about how to design the linker between two guide RNAs, we just focus on one guide RNA, and out of this guide RNA from exon 3 and 4, we focused on the where this is the guide RNA sequence, and we got the PAM sequence here. So in this sequence, we also try to check. We are going to check with respect to the CAPS assay or something like that. That's why we also need insight here. There is also one restriction site that shows CVI K1, and we can also use this site as for the CAPS assay later on. Here try to do is something like we are just generating one guide RNA here. This sequence is also for 55 bases. And the vector that we were using about the PRGB something like it has the restriction sites as a BSA1 where we can insert our guide RNA. So we thought to add this in our guide RNA at the ends. You can see the guide RNA. This is the guide RNA of the E4, and we have added this BSA1 site here. And we can also later on in the cloning strategy you will see this guide RNA will be inserted vector to be used as a knockout for the rice BADH. So later on I would like to call for saying the cloning strategy. Thank you.
So this is the cloning strategy. The vector which we are using is 32, and the cloning strategy we use is restriction enzyme digestion. Here we introduce the as you see the okay. This is our BSA1 site, and basically we produce some sticky ends, oligos, and then this is gRNA. So for cloning strategy, we basically use vectors, so appropriate adapters specific to the forward primer and reverse primer, that is included to allow the formation of cohesive ends of BSA1 sites, restriction sites that will also follow annealing by ligation. And then through the binding vector system, here we with the help of binary vector system, transform that in agrobacterium mediated transfer. So I would like to call Tanushree, which will discuss plant transformation.
So very good morning everyone. My part is to describe the transformation portion. As we all know that mediated transformation is started callus tissue culture. So we are using the Smriti Rice 1121, and we are starting with the seed inoculation. So after sterilization we
target size, we should paste the target sequence in the NAV cutter. In the NAV cutter, where there is a software where we can design site for the caps marker. And upon submission, we will get a list of the restriction enzymes that guide RNA, and then we should locate the guide RNA region in the sequence and search for the restriction enzyme that specifically based upstream of the PAM site. And this is a website of the NAHEP cutter where we will copy and paste our gene desired gene into it, where it will give the list of restriction enzyme those present here, the restriction enzymes that are present for our target gene. And this is a detailed protocol of the CAPS assay where we will amplify our gene using the PCR and using the nested primer surrounding the target SGRN region. We will subject it for restriction followed by agarose gel, and we will analyze the result of the CAPS assay. We can see here the presence indicates the wild type, where the absence of one band indicates the mutant. So we can say when the mutation has taken place, the recognition site will be altered, only one band. And the second assay that we can go for instead of CAPS assay is the Indel CAPS assay is very good to detect the small mutations. But in this case of Indel PCR assay, it is not like so. In case of Indel PCR assay, we will design the PCR primers for our gene of interest and PCR amplification of the region, target size, and we will go for gel electrophoresis, identify the non-mutants from the non-mutants. And once we identify the non-mutants, it is not like the DNA of DNA design and the PCR primers for our gene of mutation and gene of identification of the target size, and we will go for identify the non-mutants. And once we identify the non-mutants,
It is very good. You can otherwise anyway you have to go for Sanger sequencing, confirm whether mutation is there, or if any indel or any kind of. So here only one you are Sanger sequencing, confirm whether mutation is there, or if any indel or any kind of. Next one. Okay, you were saying you don't have a linker. What is that linker? What is that linker for two guide RNAs? So here only one you have a Sanger sequencing, confirm whether mutation is there, or if any indel or any. Okay, next one.
Have a polysystronic expression, so we need something that we need.
Okay, you were saying you don't have a linker. What is that linker? What is that need to have for two guide dependent, two multi-dependent, two multi. So here only one you have a Sanger sequencing. You need promoter.
We want to have a
You don't need. It is not head to head. You are saying you don't have a linker. What is that linker? What is that need has for two guide? Has for two guide dependent, two multi-dependent, two multi. So here only one Sanger sequencing. You need promoter. One guide is not a polycystic. It is not independent. You are saying you don't have a linker. What is that linker? What is that need has for two guide? Has for two guide dependent, two multi-dependent, two multi. Designing an experiment, but when you think in a broader talk, so in terms of India, export, I'm sure if you have to start with 1121, you have to take permission. Okay. Right?
Yes,
because a huge export market is there, such kind of modifications probably may not be allowed. So that you have to take permissions and then you have to do. So when you do experiments, you have to see the repercussions or a larger view also. So in lab, just for designing, yes, good, but when you get into practical aspect of doing these things, so you have to take care of those things.
Ma'am, that's why we took name of Pusa Basmati 1121 because it has been like replaced by Pusa Basmati 1885 right
but
now.
It's still,
It's
still, okay? Because everything
that will take help of EKISing, sir, ma'am.
Yeah, not EKISing. Otherwise, also bodies are there. Government bodies are there. They allow and all that. So, when you do such things, you have to take care of those things also, permissions and everything. Next one. Okay, fine. Yeah, you don't need not go for NAB again, right? So you can do that's fine. Yeah, okay. That's fine. Rest is fine. Huh? Okay.
Thank you, ma'am. Thank you, sir.
So we have completed presentation by 3 groups, and rest of the 2 groups will be presenting later. So before going into the next presentation, I would like to thank Dr. Monica ma'am for very nicely evaluating both the all the proposals so far, and we would like to felicitate her. For that, I would like to request Dr. Bhattacharya sir for felicitating them. And now there is a little change in the program. The next talk is by Dr. Bhattacharya. As you know, he is director of NIPB, and he will be talking about a very important aspect of genome editing. That what are the standard operating procedures. So DBT has come up with standard operating procedures in October 2022, and he will be briefing about those procedures. So sir, you are please invite.
So good morning, lecture after lecture, but I hope you are finding all the lectures very useful. So in this session, the purpose is to tell you about the regulatory framework that has been notified to handle the genome edited plants in India, and all the procedure or protocols that one has to follow is decided by Department of Regulatory Framework, Ministry of Science and Technology, that has been notified. So here to handle the genome edited. Purpose of this lecture is to tell you about the so-called SOPs that is notified by DBT for following in case of developing genome edited plants, and this SOP is available on the DBT website, so you can visit that website and you can go through that document to find further details. But nevertheless, I have tried to take salient features of that document and project in front of you, and explaining to you what does it really mean, and how one has to adhere to those guidelines. So before I go into the actual content of this class, I just want to emphasize that new thing has come because of development of this genome editing procedures. If you look into the milestones in crop improvement program or plant breeding program, you will see that crop improvement program has actually started since 10,000 BC, when we first did the domestication of wheat. So when the human being settled into agriculture, they tried to look into plants which are suitable to give them food. So in that effort, they looked into the natural variability of different species, and then they picked up few plants which were seemingly suitable to fulfill the need for food in settled agriculture. And then that effort, the science began in this variability in this plant-breeding discipline. As and when there is a significant development in biological science, immediately it has been impregnated into the crop improvement program, to devise various methods, various strategies. Ultimate aim is to produce more food, and more desirable food. And in that journey, significant development is the crossing technology or hybridization technology, where after learning from Mendel's law or Mendel's finding, we understood that one can convert different characters by crossing two different plants, if crossable, and you can bring in the traits from two different plants into the progeny by making hybrids or by crossing them, and that gave birth to the hybrid breeding technology. And then we also realized that if you properly choose the right kind of parents before crossing, your F1s are superior than the parents. We called it hybrid vigor. And then a lot of hybrid varieties subsequently developed, and so on. But very soon we realized that we are ultimately dependent on the kind of variability that is present in the nature. So the immediate question was that, can we create new kind of variability? And for that, way back in 40s, we also learned the mutation phenomenon, how the DNA sequence can be changed by the use of various mutagenic agent, physical agent, chemical agent, and then people started mutation breeding. They expose the seeds to the mutagens, various kind of mutagens. You know that EMS, MMS, chemical mutagens, and then UV ray, gamma irradiation, and so on. And then they created various types of changes in the genetic material with the hope that new kind of variability will come, and from there a new selection would be possible to do to further improve various traits for which otherwise the variability was very limited in the nature. So you have many desirable traits for which you don't have variability, so how do you improve them? The only way is you create variability, you create desirable type, and then you take it forward by selection and other things to improve that trait. But then in mutation breeding we faced several drawbacks. Like it was a kind of gross treatment to the whole genome, and you cannot control treatment to the particular gene or particular region of the chromosome where from you want the new variability to come. So because of this gross treatment, there will be many collateral damage in the genome. You may get a desirable changes, but then it will be associated with many collateral damage which are not desirable, and then segregating out from those negative undesirable changes the particular change that is desirable was very difficult. You need to grow a large number of population and then look for a large number of segregating plants, and from there you select, and then fixation of that new trait that has come up because of mutation was also very cumbersome. So it became very cost intensive, and because of not having much precise control over the generation of new variability through mutation, the mutation breeding could not be practiced in majority of crops and in majority of institutes. Wherever land was scarce, labor was scarce, funding was scarce, then mutation breeding could not be practiced. But then down the lane with the discovery of DNA model and the molecular nature of genetic material with the great Watson-Crick discovery, and then subsequently the gene-protein correlation by Janowski, and then how gene functions, the central dogma by Crick. I am sure at some point of time you have read those great findings of biology. So we understood that how gene functions, and we also could correlate that the particular region of the DNA molecule which is hosted in the chromosome is ultimately responsible for a trait. And then with the discovery of restriction enzyme and recombinant DNA technology, it was possible to isolate the gene as a discrete molecule and then transfer those gene for transfer a particular trait in a very precise way, and that gave birth of the transgenic technology. In transgenic technology, the first crop that was developed is the insect resistant cotton, which was commercialized, which was brought into cultivation in late 90s, and that was the beginning of another era of precision crop improvement and transgenic technology, you can say. Along with that, again, when once you understood the nature of genes, the nature of DNA sequences, and then many more methods which use DNA as a substrate, we developed the various various technology to differentiate between the DNA molecules in terms of outcome of a particular reaction. So, say for example, in RFLP, restriction fragment length polymorphism, what we do, we try to correlate the presence or absence of a restriction site. Restriction site means a particular sequence difference. And then we try to produce into a visual effect. Visual effect means you ultimately produce a X-ray chromatogram where a particular segment of DNA behave in a different way to give you different length of fragments we called restriction length polymorphism. So the difference in the sequence level is manifested into a visual pattern. So it's a one kind of gene to phenotype, but here phenotype meaning is experimental phenotype or experimental outcome. Okay, so the limitation of not having sufficient markers linked to important traits could be partially overcome by development of molecular markers. So in traditional breeding, whenever you want to exercise a selection for a particular trait, how do you do that? You essentially need a marker, visually detectable marker, through which you search the plants. Right, suppose for example, you found that whenever the plant is insect resistant, it has trichomes. It has trichomes in the apical meristem or in the leaf petiole. So that presence of trichome becomes your morphological marker, and then based on that you can select insect resistant plant. But if this kind of morphological marker is not present, it becomes very difficult to select the desirable trait in traditional breeding, because then what you have to do, you have to go for assay, release the insect, see which plant is damaged, which is not damaged, and that becomes very, very cumbersome. So that kind of problem was overcome by the development of molecular markers, where once a molecular marker is developed, then it can be practiced, it can be used, and that gave birth of the marker assisted selection or marker assisted breeding, MAB or MAS, right? So all these developments happened towards the end of the previous century, and then we started calling that this is the era of precision breeding. We entered into the era of precision breeding, where precisely we can follow or track a particular trait or the gene segment or the DNA segment responsible for that trait by following the molecular marker or by using the molecular marker. So that is the kind of precision breeding. And another part of precision breeding is that by transgenic technology you can incorporate a gene and modify a trait without altering any other trait or feature of that particular variety in a very, very specific and precise manner, right? So that was the era of precision breeding. But then in this precision breeding, it was mostly all kind of convergence that was possible. Means you can introduce a gene by transgenic development, you can bring in a set of genes by hybridization, you can bring in a gene from a wild species by gene integration or wild hybridization, and so on. But then what was missing, that whenever you required to eliminate something, then it was very difficult. You were still dependent on mutating that gene, which was very, very cumbersome. So that part was taken care of by the development of various methods, one after another. The first one is, you know, that where you can silence a gene or you can reduce its expression through RNA interference. By introducing a double-stranded RNA molecule for a particular sequence or for a homologous sequence, you can attenuate or you can interfere or you can reduce the expression of that gene. And you know, I assume that you know how the RNA interference works, how the transcript is degraded, and then you don't get the protein for a particular transcript, and we say that it's a kind of gene silencing, and then thereby you stop the expression of a particular trait which is undesirable for you, right? But that was not a sort of, you know, a foolproof system, because most of this RNA interference work has shown that you can reduce but you can't really completely knock out, because the efficacy of the system is so that it won't, in most of the cases it would not give you the 100% silencing of a gene. And then very recently, although it's not very recent, it's more than a decade now, the various methods of gene editing has come up, where precisely you can knock out a gene. You can create a mutation at a particular site, at a particular specific target site. So specific mutation or targeted mutation through genome editing, and then in this various important or significant milestones that are that led to the evolution of this genome editing technology as a very user friendly and universal method which can be followed by any of the labs throughout the world. That has been told in the beginning of this lecture. I hope that what are the significant developments which led to the refinement of these genome editing techniques to be practiced in lab for the purpose of crop improvement. So I am not going into that, but what is important is that after the development of these genome editing techniques, now both the sides are accessible. In the sense, you see here, I am tried to show you the picture of a globe, that if half of it is day, half of it is night. So far all this precision breeding was accessible for one particular thing, that was convergence breeding. But after the development of this gene editing techniques, now the other part is also accessible, the elimination breeding. You can selectively eliminate or modify any trait by creating very precise mutation in the target region. So again, the context of today's class is to really compare that why we are so enthusiastic about the genome editing techniques. What was not there, now it is there. So one thing is the technological advantage or technical advantage in terms of giving you ability to eliminate some gene, not only adding gene but eliminate but gene. But then it is all the more interesting, or I would say very exciting to the scientific community, because of the regulatory framework that has been decided by the government of India with a lot of liberty, with proper scientific explanation, that whatever difficulty we face in case of handling the transgenic product of transgenic plants, some of them, those obstacles would not come when the product is developed by genome editing. That is very important. So the usual procedure to develop a transgenic plant or to undertake a research related to the development of transgenic plant is that once you decide to develop a transgenic plant, the first thing that you have to do is you have to submit your application to the institutional biosafety committee. Every institute must have an institutional biosafety committee, and then how that committee will be constituted, there is a prescribed rule which is given in the DBT website, that how to constitute the institutional biosafety committee, what will be the composition, who will be the members of that institutional biosafety committee, and it is given in the website. There is a IKBP portal. One can go and see. So every institute which undertakes this kind of research must formulate a IBSc, and then the applicant will go to the IBSc with all details of the project that the applicant wants to start, and this is right before beginning of the project. And then IBSc will review that what is your work and whether it is important or not, how the experimental steps will be handled, whether the institute has proper infrastructure facility or not. After considering all these aspects, IBSc will give you approval or may not give you approval. So once you get approval, parallelly IBSc will send all this important information to RCGM, that is again a body constituted by Department of Biotechnology. That RCGM will be only informed at this point of time. They do not have much role in giving you the approval, but IBSc has authority to give you approval for beginning of the work. But then as you develop the plant, then again you have to go back to the RCGM with all details. RCGM will look into the results, and then they will put up your case for approval for a trial. We called it confined field trial or CFT, right? So this is the first time that you are growing your plant outside the lab, but in a sort of confined condition, in a restricted area. Okay, so it is not that you can grow anywhere. There will be a designated area which will be isolated from so many parameters are there how to designate an area for confined field trial. So in that designated area, you will be given permission to start your trial, confined field trial or CFTs, and then these CFTs can be of two scales. The first one is called BRL1 and the second one is called BRL2. I will come into that, what is BRL1 and BRL2, but before that at the same time RCGM will constitute a central compliance committee which is called CCC, and again in that committee there will be several experts. There will be state government officials, there will be central government officials like from DBT, there will be subject matter specialists and many expert members. That committee will be constituted by DBT, and they will send that committee to visit your trial site to see that whether everything is proper as per guideline or not. Okay. Then once you satisfactorily complete the CFT or BRL1, then your result will be put up to all the CFTs, BRL1 and BRL2, then your result will be put up to GESE, Genetic Engineering Approval Committee, and then they will look into the data, they will see the promise of the material, and they will consult with the stakeholders. Stakeholder means whomsoever is positively or negatively affected by this product. Say for example, farmers will be the stakeholder because they are going to be benefited. Say for example, any environmentalist could be a stakeholder because they may raise concern that it has some negative effect or so on, if at all it is there. So all possible stakeholders will be consulted, experts will be consulted, it will be put up in the website for all stakeholders to go through the data, to go through the result, to analyze the pros and cons, and then after getting the feedback from the stakeholders, the GESE will make a final decision and may approve or may not approve the environmental release of that transgenic material or release of that material for use by the farmers. So the procedure seemed to be very lengthy, and procedure seemed to be a rigorous exercise required by the scientists to do. And most of the time, since it involves several steps of approval process, scientists were not very comfortable to pursue those steps because they are scientists, they would not be very good at following up or pursuing through these steps. So many a times it so happened that even if the product is very good, it did not see light of the day because of all these lengthy and cumbersome procedures which are essential to deal with any sort of transgenic materials. So as I was telling to you, first of all, why field trial is important? Because this is the first time you are growing your plants in the natural environment, but that field trials will be confined. Why confined? Because the biosafety analysis or analysis of other data is still under process. So therefore it is confined. You are not allowed to grow in a very widespread manner, but in a designated place in a very confined way. Therefore it is called confined field trial. So primarily CFTs are conducted for event selection. Event selection means you have developed multiple number of transgenics. Now some transgenics will be good in expressing the trait, some may not be satisfactory. So that is called event selection trial. Which event is very good or manifesting the trait in a very effective way. So that is the one purpose. Second purpose is to collect material for other analysis related to biosafety. Then experimental seed production. You can produce seed and anticipating that next steps will be trial at multi locations, means at different agroclimatic zones of the country or different agroclimatic conditions of the country. So because of all these various reasons, the confined field trials are conducted, and the difference between the BRL1 and BRL2 is in terms of scale, means how much area you will grow. You can see here the values are given that BRL1 is limited to not more than 1 acre per trial site and maximum cumulative total of 20 acres. Whereas in BRL2 it will be 2.5 acre per trial site. So it's a little bit scaling up, growing of the plant and so on. So this was the usual procedure that one has to follow in case of handling the transgenic events or transgenic material. But when you do the genome editing, particularly using CRISPR-Cas method, you have seen that the advantage of this method is that the machinery that does the mutation, they all are transacting. Transacting means as where the changes are brought, the machinery are not present there. They are present elsewhere. They may be integrated into the chromosome, or they sometimes do the mutation without being integrated. That I am sure you have gone through the basics, that how CRISPR-Cas works. So since they are transacting, so after the mutation is created, you can separate that out. You may not take them, you can go ahead only taking the mutation. That is the advantage. So mutation is created, but all your CRISPR construct, CRISPR RNA, Cas9, whatever you have used, they may not be part of that product. That is the advantage. Considering that, and you I am sure you know how it works, I am not going into detail, but the important thing is that there could be three possible outcome by that can be created by using CRISPR-Cas methods, and they are designated as SDN1 type changes, SDN2 type changes and SDN3 type changes. And again I expect you understand by this time that what is SDN1, what is SDN2 and what is SDN3. Fine. So SDN3, what is that? What is SDN3 type of changes? No murmuring, you tell clearly. SDN3 means where you are integrating something, some new gene or new sequence using this technology, that is SDN3, whereas SDN1 and SDN2 you do some mutations in a minor way. either by mostly by non-homologous end joining as a outcome of non-homologous end joining or repair mechanism. It may be point mutation, it may be small deletion, or it may be some targeted changes from one nucleotide to another nucleotide, right. So, this is the primary logic is that, or the primary principle in differentiating SDN1, SDN2 and SDN3 is that SDN1 and SDN2, since the changes are small, so one can assume that those kind of changes are already present in nature. You know that in nature, for any gene there could be several forms, we call it allele, right, and those alleles differ in this sort of differences, means there will be some nucleotides changes if you compare any two alleles or among the alleles. So, with that logic, SDN1 and SDN2 are considered as one category, SDN3 is a different category, and then Government of India, they issued a OM saying that if it is a SDN1 and SDN2 category of changes, they will be exempted from rules 7 to 11. So, not all the rules, but rules 7 to 11 that needs to be followed in case of transgenic material. So, there is a set of guideline which is commonly known as rules 1989. That guideline is followed for handling transgenic material before giving the environmental clearance. So, this OM says that from that set of guidelines, rule 7 to 11, the genome edited plants are exempted from rule 7 to 11 of those guidelines which are prescribed for recombinant material or recombinant or genetically engineered plants. So, let us see what were those rules 7 to 11. Exemption from rule 7 to 11, how did it help or how it will really simplify the things. So, the OM indicates that the development of genome edited plants will be carried out under containment until free from exogenous introduced DNA. So, all the development procedure needs to be carried out under containment condition. It has to be within a lab which is certified to carry out recombinant DNA technology procedure. In between, if you want to grow plant, it has to be in a designated place like say glass house or growth chamber which are designated or allowed to grow the recombinant plants or genetically engineered plants. So, all this developmental procedure has to be carried out under containment until the plant is proven to be free from exogenous DNA. Exogenous DNA means whatever you have introduced to do the CRISPR-Cas technology, so called sgRNA, Cas9, all those things. Second thing is, for such genome edited plants, once it is proven that it is free from exogenous introduced DNA, once it is proven, then it will be treated as a normal bred variety, and for varietal release it will the existing procedure will be followed. So, the way a traditionally bred variety will be handled for evaluating it before release to the farmers, in the same way it will be handled once you prove that it is free from exogenous DNA, and that certification will be given by Institutional Biosafety Committee, IBSC. They will analyze and they will give you the certification. Now, in this SOP, there is a clear definition that what does it mean to say SDN1, what does it mean to say SDN2. It is very important to understand that what exactly is SDN1, why it is exempted from rule 7 to 11, and what is SDN2, why it is exempted from rule 7 to 11. Most importantly, once you know what is SDN and SDN2, you see the last 2-3 lines. These resultant mutations are comparable to those occurring in nature, obtained through conventional mutagenic treatments or natural variation found in the primary oblique secondary gene pool. So they say this could be otherwise present also, and therefore exemption is being given. Understand? So the kind of changes that comes under SDN1 type, they are being given exemption because of this logic. Similarly, for SDN2, which involves a template guided repair of a targeted DNA break using an externally supplied template sequence, but at the end of the day the changes are minor. The resultant mutant carries a modified sequence leading to an altered expression profile of the gene and or altered activity of the encoded protein or RNA, and thus the edited version could be regarded as an allelic form comparable to those available in the primary or secondary gene pool. So they are being exempted because the basic assumption is these kind of changes may occur or continuously occurring in nature also, and because of which you get multiple alleles. Now once you learn about the SNPs, you know that there could be so many numbers of allelic forms, as many as the number of nucleotide in a particular sequence, isn't it? Plus some addition, deletion still may happen. So with this logic, the exemption is given from rule number 7 to 11. Now what is that rule number 7 to 11? What did you really get in exemption? Rule number 7 describes all the prohibition that are imposed on a genetically engineered variety. What are the kind of prohibition that is applied in our country when it is genetically modified or GMOs, genetically modified organism? The first thing is on import, export, transport, manufacture, sale of genetically engineered organisms are prohibited except with the approval of the GEAC, Genetic Engineering Approval Committee. So until and unless you submit your case, GEAC gives you approval, you cannot import, export, transport, manufacture, process or do any activity or sell a GMO. Second thing is, again, handling of this kind of GMOs shall only be allowed in laboratories or inside laboratory area notified by the Ministry of Environment and Forest for this purpose under the Environment Protection Act 1986. So until and unless your lab is notified or until and unless a particular setup is notified, you are not supposed to do anything with the GMO in that setup. Okay. Then the GEAC shall give directions to the occupier to determine or take measures concerning the discharge of genetically engineered organisms or cells mentioned in the schedule from the laboratories, hospital and other areas, including prohibition of such discharges and laying down the measures to be taken to prevent such discharges. Means you cannot give it to anybody. If you want to give it even from a certified lab to another certified lab, there is a permission process involved. I just cannot say that I will give it to you because your lab is also certified, so what is the problem? So I have to write to the authority that I want to transfer this GMO from, say for example, one of the institute's lab to say one of the lab in ICGB or JNU. So I have to take permission and then I can give. Any person operating or using genetically modified organisms, if they want to scale up or do any pilot operation, shall have to obtain license issued by the Genetic Engineering Approval Committee. Suppose I developed a transgenic plant and I want to multiply, I want to do seed production. I would say, why should I need permission? I can do myself because I am not giving it to farmers, not giving it to any stakeholder. But no, then also if you, since you are scaling it up, you are growing in a larger scale, so you have to have permission. So in one way you can say anything with the GMO you need permission from GEAC. So that was the sort of requirement, and because of which the development sometime becomes very slow. because for every step, until and unless the permission is obtained in a timely manner, you cannot proceed. Certain experiments for the purpose of education within the field of gene technology can be carried out outside the laboratories and laboratory areas mentioned or designated, that will be looked after by the IBSC. So, in laboratory and also in some annexure kind of thing, associated facilities, infrastructure facilities, whether GMOs can be taken there, grown there, it will be, permission will be given by the institutional biosafety committee. They will see, consider all different aspects, and they will give you permission, and once permission is given, then only you are allowed to do that. Okay, so again for production also same, one has to take permission from GESE for any production, otherwise it is prohibited. So you see this rules basically same is for, I am not going through all line by line details. Deliberate or unintentional release, how to prevent it, if someone does, it is punishable, so all these things are there. Permission and approval for certain substances, products which contain genetically engineered organism are not allowed to produce or be sold or imported without the permission of GESE. Even for food stuff also, until and unless it is permitted, you cannot use it if there is a chance of that food stuff containing GMOs. So this rule 7 to 11 has taken care of that without permission you cannot do all these activities with any GMOs. So from this rule 7 to 11, the genome edited plants are exempted. You understand? They are exempted. That means what? That means you can do all these things with the permission of only institutional biosafety committee. So institutional biosafety committee has given authority to give you permission, and that in fact hastened the process, because institutional biosafety committee is right at your institution. At any point of time you can approach, you can seek permission. The process will be very fast. The process will be very fast, and then once it is proven that it is free from exogenous DNA, then it will be treated as a normally bred variety. Okay. So that is the kind of enthusiasm that has come. Now how the whole procedure works. When you initiate the research, right at the beginning you have to submit your application in a specified application form, which is called D1. The D1 form basically asks every details about your plan or about your project, that which gene you are going to mutate, how you are going to mutate, how will take care of the progeny, which vector you will use, what is the sequence of the vector that you are going to use, what is the sequence of the guide. And so all experimental details you have to fill up in D1 and then submit it to institutional biosafety committee. They will go through, they will see that whether it is important to take that kind of research, is it going to give you substantial advantage if you achieve this kind of research objective. Considering everything, they will advise you, and at the same time they will approve beginning of the experimentation. Then you start your experimentation, but then in the experimental procedure, while carrying out the experimental procedure, you are under the obligation of regulations and guidelines for recombinant DNA research and biocontainment 2017. So it says you the do's and don'ts. So you are under the obligation to follow this guideline. All these guidelines are available through DBT website, so you can go through in detail. Applicants and IBSCs has also referred to the handbook for institutional biosafety committee by DBT, which is updated periodically. You can look into the IBSC guideline also. And then IBSC will keep on informing RCGM for information through IBSC minutes, means whatever meeting IBSC do with while considering your case, that proceedings will be sent to RCGM, and RCGM shall communicate his observation to the applicant and IBSC. So there is a continuous dialogue between the central government agency and the institutional biosafety committee, but institutional biosafety committee is given authority to approve your case. Now there is a 3.2 section says suggested procedure for handling genome edited plants. Again, you know, there is a clear cut instruction. The advantage of this SOP is that it is not left on the experimenter. Sometimes experimenters, they are baffled that how to really prove a particular thing which is acceptable to the authority. But here you are given a clear detailed procedure for each and everything. That is the advantage. Say for example, how will you handle the progenies? Progenies of individual T1 events should be maintained separately. Self seeds of individual T1 plants should be raised in a single plant to progeny row. See, every detail is given. That how will you grow the progeny plants, how will you do the assay. Investigators are recommended to provide the following information to the IBSC. Number 1, number of T0 transformants generated, the generation in which mutation is detected, whether in that generation mutation was detected in homozygous condition or heterozygous condition, whether mutation will be maintained as heterozygous or homozygous condition, if homozygosity has attained, in which generation it was attained. So all these things you have to record, you have to do the experiment and record, and this information has to go to institutional biopsy committee. And then generation at which genome edited lines were found to be free from exogenous DNA. So once you developed, in which generation you found that they are separated or they are segregated from the exogenous DNA. The IBSC regularly monitor the activities as per the handbook of the IBSC given in the DBT portal. And then once you seek exemption of that 7 to 11 rules of 1989, what are the information you have to provide? Then IBSC will consider to give you exemption. That is the sort of final permission. Once you get exemption, you are free to take it for trial in a normal way. So for seeking exemption, you have to provide all this information, as I was telling to you, that which category of genome editing it is, SDN1 or SDN2, then confirmation, whether you have confirmed that what kind of addition, deletion, change in nucleotide has happened with DNA sequencing, whether selfing or backcrossing has been carried out to segregate the exogenous DNA, whether by selfing you have got a line free from exogenous DNA or you have done backcrossing for that, you have to mention, whether any DNA free method has been used while developing the plant. DNA free method means RNP. You might have been told RNP method, that instead of doing a transformation, sometime you can use the Cas9 protein and sgRNA as a RNP, ribonucleoprotein, and do the medical. So there is no permanent integration. So there is no question of exogenous DNA integrated into the genome. And that is particularly important when you handle any tree species, because you know tree, if it is a perennial tree, how do you get several generations of that tree? It becomes very long time taking procedure. So in those situations, the RNP is preferred, because there you do not have option to look into the segregating population and go for a plant which is free from exogenous DNA. So whether any RNP procedure has been followed, then evidence to confirm that the genome edited plant is free from exogenous DNA. You have to give evidence proving that it is not containing any of the tDNA portion that has been used in developing this plant through using a binary vector, having CRISPR, sgRNA, all those things. So how will you prove? What are the kind of evidence? That is again very clearly mentioned, that how you can prove whether your plant is free from exogenous DNA or not. I will come into that, but before that, depending on the type of modification and trait expression under containment condition, phenotypic data to be recorded and provided. Phenotypic data, how is the yield, how is the growth of the plant, branching, length, everything, whether they are resembling the normal plant or it is a new altogether different plant type has come up. So all those phenotypic data has to be recorded. In case of any unintended phenotypic changes, whether it was selected or segregated out. Suppose you got some off target changes. So have you preferred to take it along or you have segregated out? That has also needs to be mentioned. In case of nutrition related modification, you have to compare with the non-edited version, that what suppose you have done some genome editing to increase the iron content in the seed. So you have to compare with normal seed with this genome edited or modified, and then you have to show that how much advantage has come. So compare with the normal counterpart and show the advantage that has come because of this modification. Whether information on identical allele is already documented, if yes, information to be provided. So many a times that particular allele is already present in nature. For example, just a hypothetical example, you might have heard about BLB resistance. Bacterial leaf blight resistance is governed by some recessive genes, isn't it? So XA21 or those kind of genes I may not know in detail. So you know that this kind of changes in the wild type allele will create a recessive allele and the plant will become resistant. So traditionally what you do, you have some lines with those recessive mutation, you will start backcrossing and transfer that trait over the generation. But having advantage of this CRISPR-Cas, now what you can do? You can do similar kind of mutation or changes in a allele in an existing leading variety. That will save the time, plus that will save you from all the disadvantages that are associated with introgression breeding or backcross breeding, right? So just precisely you create that mutation. So there is no question of additional undesirable things coming, no question of genetic load coming. So that is the advantage. So if that natural variant is already occurring, but you have done through CRISPR-Cas, that has to be also maintained, and that's become a very, very advantageous, or you know, it support your case. That is already there. I have just created the similar mutation in this leading variety. So that is the whether information on identical allele is already documented. If yes, please give the information. Now applicant shall use protocol or methods prescribed in section 5 of this document to show that the genome edited plants are free from exogenous DNA. So a protocol is given to check and show that whether your plant is free from exogenous DNA or not. Before I go into that, there are other things specified here, that all this information to be provided in specified format, annexure 2, annexure 3, and they will be examined by the IBSC. If found satisfactory, they will give you the exemption, and that exemption, while conveying to you, again there is a specified language to give you that certificate. In case methods other than specified in the SOP, if you have used to prove that your material is free from exogenous DNA, that will also be considered, and if the committee is convinced, it is allowed. So it is not that you have to use their procedure or specified procedure. You can use any other modern method, but it has to be convincing. The committee has to be convinced that okay, this method is acceptable to prove that your plant is free from exogenous DNA. Further development, evaluation and release of the genome edited plant as a new variety or hybrid shall be governed as per other applicable laws. Means as a normal bred variety, it goes into trial, multi-location trial, variety release committee's consideration. So same procedure will be subsequently follows. In case where the genome edited plants does not fall under the categories of SDN1 and SDN2, then it will be treated as GMO. Means if by any chance, if you have introduced a new gene or introduced a new sequence, or your some part of exogenous DNA is still present, that means it is not SDN1 or SDN2. It goes into SDN3 category, and it will be treated as transgenic material, and you have to follow all the rules that are need to be followed by for recombinant DNA material or GMO. So again, for import export, similar kind of rules, similar kind of information to be provided to the IBSC. They will look into it. So suppose you are importing a plant which is genome edited, and your experimental plan is to use that line for subsequent transfer into your choice of cultivars. So that also will be, permission will be coming through IBSC. So you have to give all details that such and such plant developed by such and such institution, I want to procure, I want to use it as one of the parents in making such and such crosses. That has also be, all details has to be given to IBSC, and then it will be taken care of, or permission will be given. If you are importing from any lab outside the country, then existing quarantine regulation will be applicable. Okay. For importing anything, not only any, nothing to do with genome editing or GMO, for importing anything, it has to go through a quarantine law, which is taken care of by NBPGR, National Bureau of Plant Genetic Resource, here our sister institute. So there, in case of genome edited plant also, if it is coming from outside the country, it has to go through those quarantine regulatory laws. They will check presence of any unwanted things, any fungus, pathogen, weed, etc., etc. So it has to go through. Okay. Then it says these SOPs are applicable for the import of genome edited plants, seeds, propagules and other product for research, testing and product development only, and not applicable for trade and commerce. So it is all together for research and development, these SOPs, not for selling. For selling, it has a several, it has a separate guideline to follow. It has nothing to do with this SOP. Okay, it's not for trading. Now, as I said, to put it in a nutshell, the overall procedure, that you fill up the form D1, submit it to IBSC, IBSC review the case, and they instruct you to carry out all the activities in contained facilities, and then they give you permission, and then IBSC inform the RCGM that such and such research is being carried out by such and such person in such and such institute, and then when you carry out all the activities, there will be regular reporting to IBSC, and then once your product is developed, then you apply for exemption. Okay. IBSC report as information to RCGM by submitting minutes of the meeting through the portal system IBKP. Through this portal, IBSC will keep on updating the RCGM that what is happening and what is the development, and then further evaluation and product development as per other applications. So once you are given the exemption, then you enter into the protocol which is followed for any followed for release of any variety, even if it is bred traditionally. Now, how do you prove that your material is free from exogenous DNA? There is again a set of procedure. For example, one has to perform overlapping PCR or nested PCR. So, what is the part that is transferred into your genome edited plant while developing the mutation? The tDNA part of the binary vector, right? So, over the entire span of tDNA, you have to design primers in a overlapping manner so that every amplification will be within 500 base pair. Understand? So, first 500 base pair you design primer, then in overlapping fashion another pair of primer, 500 base pair, then again overlapping fashion next 500. So, entire span of the tDNA you have to develop primers, you have to do PCR amplification, and you have to prove that no amplification is coming. Okay, no amplification is coming, and while showing that no amplification is happening, and because of that I claim that there is no exogenous DNA, you have to show the result with proper control. Otherwise, you might have done some mistake in your reaction and you are showing no amplification has come. That's not allowed. So, you have to take proper control. What is the control? For each amplification reaction, 3 PCR positive control must be used. Number 1, a well known endogenous low copy gene, okay, of the same species using the same amount of genomic DNA sample of the final edited lines. So, under similar conditions, same amount of template and all the reactions are done in same way, you have to do a PCR taking endogenous gene which are commonly present. We call housekeeping genes, right. This is one control. Second control is the genomic DNA of a segregant line which is harboring the exogenous DNA. You understand? Means genome edited plant, but still it is carrying the exogenous DNA. That will be another control. So, their amplification should occur. So, that will be another kind of positive control. And third control is the parental genotype spiked with 1,000th of the full length purified vector DNA. So, the parent genotype where you have started your experiments, that has to be spiked with the vector construct DNA that you are using, and then you show that amplification is happening or not. So, in all these positive control, amplification should happen, but if it fails, that means you are doing some mistake in the reaction, your result will not be acceptable. So, these are kind of positive control. The amount of template DNA and the PCR conditions should be such that a clear band of expected site is visible in all the positive control, and no amplification should be detected on the DNA for which you are claiming it is free from exogenous DNA. Okay. And then if you use any other method to prove that your plant is free from exogenous DNA, that will also be considered, provided the members of the IBSC are convinced about applicability of that method for your case. Okay. So, it is open, it is not that only this procedure has to be followed, it is open, but then they have to be see the reasonability. Now, to bring the uniformity of the plants which are developed through this technology throughout the nation, right at the beginning they have given a method for nomenclature of these genome edited plants, and this nomenclature says that the first 2 letters of the genus followed by the first 2 letters of the species or variety name or accession number, then UAC number of D1 form, your application form's reference number, and then event number. Examples are given below. Suppose for a mutation of chickpea, Cicer aureatinum, accession number ICC 4958, UAC number of D1 form is whatever number is given, event number 1, the name will be as first 2 letter of genus CI, then next 2 letter of species aureatinum AR, then the accession number, and then your D1 form reference number, and then event number. So, this is the standard way of nomenclature of any genome edited line for and then another example is given. Okay. So, and then this SOP will be open to modification depending on the development in the subject area. So, if some new thing comes, new method comes, all these things will be considered, and accordingly this SOP will be further modified, particularly in the methods to be used to show that the genome edited plants are free from exogenous introduced. So, it is very essential to prove that your material is free from exogenous DNA, because based on that only the all exemption is being given. So, one has to be very, very particular and flawless in proving that the material is not having any exogenous DNA, and if there is any revision in the SOP, it will be posted in the DBT website for information and compliance by the entire country. Okay, so, this is the overall SOP, and I am sure that it does not sound much complicated, it is pretty simple. Provided your plants are good, then there is all sort of enthusiasm around to support you to take that advantage to the stakeholders. Okay, so I'll stop here. Thank you very much for your patient learning, and you may ask any questions if you want, or otherwise also you can ask later, no problem.
Hello sir, thank you for such presentation. It's very necessary for us. And my question is, you have told about the RNP method. So what will be the fate of this Cas9? So is it also an exogenous protein?
Yeah, so in RNP method, basically you are delivering, so for this CRISPR-Cas mediated genome editing, primarily you need two transacting molecule, right?
Yes.
One is the single guide RNA that is composed of two parts. One is CRISPR RNA, another is transactivating CRISPR RNA. So single guide RNA, one molecule, and then Cas9 protein.
Yes.
Right. So this Cas9 protein and that single guide RNA is complex, that is called RNP. It could be complexed on a particular matrix, or there are many different ways, and then you deliver it inside the cell. So there is no question of getting integrated into the chromosome, because you are delivering the enzyme, you are delivering the sgRNA, so it does the job. sgRNA goes and binds somewhere, Cas enzyme goes and cleave that and do everything, and then mutation is created. But at no point of time these things are integrated into the genome. So that is the difference. So there is no difficulty.
So there is no need to make our plant Cas9 free.
At any point of time they are not transgenic.
Okay, so
otherwise if you use a binary vector the way you presented in the morning, so there for some duration it is transgenic.
Yes.
And then once mutation is created, since it is elsewhere, you are segregated out. Understand?
So there will be no penalty for
no
Cas9.
need of segregating out, because
Inside
you are
of
not allowing
segregation.
integration.
Yes.
So no need of removing it. That is the difference.
Okay. Thank you, sir.
Sir, I am working in Trivandrum, where I have developed my construct in agrobacterium, and I want to continue my work in Trichy. There is tissue culture material, embryogenic cell suspension. For transferring that construct from Trivandrum to Trichy, we need permission.
Of course, you have to inform your institutional biosafety committee, and they will give you permission.
Institutional biosafety, it will be in our institute level only.
It will be in your institute. See, if the institute is doing any recombinant DNA work,
yes sir, already we have permission for
it must have a IBSC. So there you have to write that because of this purpose you want to carry this plasmid from this place to this place. They will give you the permission.
Sir, other than institutional biosafety, any for traveling, in that case anything?
No,
permission?
otherwise nothing is needed.
Okay sir. Thank you sir.
Good.
So if there is no other question.
Excuse me, sir.
Yeah, please.
Yes, please.
I have put some questions in the chat box, sir. Can you please answer those questions, sir?
What is your name?
My name is Abhishek, sir.
So can you specify the question, because there are so many chats.
Yes, sir.
Yeah,
I can,
please.
sir. Sir, does the same SOP is applicable to RNPs, or else is there are any exemptions to the RNPs, sir?
No, same SOP is applicable, because right in the SOP they will be asking you whether you have used RNP method. Okay, but then also you have to show that it is there is no exogenous DNA whatever you have used. Since logically you don't get any integration, so naturally it will be easily proven, but you have to prove. It cannot be left on assumption that since I have used RNP, so I need not to show it. No, it is not like that. You have to mention that you have used RNP and you checked as expected, there is no amplification, so it is free from exogenous DM.
Sir, and is there any extra biosafety protocols to be followed for nano-based RNP transfer? Because the transcription using nano-based RNPs.
No, whether it is, see, any delivery method, whether it is nano-based or any other method, it will all come under the same guideline. Because it's a delivery method which may differ depending on a particular technology that you may use. That's okay, but then you fall under this guideline if it is a genome edited plant or plant developed by application of genome editing at any point of time.
Okay, sir. Thank you, sir.
Okay, so इसको कर दूं?
Yeah. So thank you, sir, for nicely elaborating all the SOPs. I am sure all the participants will be highly benefited, and it is my pleasure to felicitate Dr. Bhattacharya sir.
Thank you
for his lecture.
So one group has to present, I have been informed. Which is that group? Is it Arjun? Arjun has presented. Nakul and Sahadev are left. Sahadev is not here. Okay. But Nakul is ready to go, right? Abhi tea break karna hai? Acha. So tea break, yes, why not?
We will resume after tea break.